Today we will study about somaclonal variation. What do you mean by somaclonal variation? Details about somaclonal variation. Let's begin. The genetic variation found in the in vitro cultured cells are collectively referred as somaclonal variations. The plants derived from such cells is referred as somaclones. Some authors use the term calicons Cali- caliclones and protoclones to represent cultures obtained from callus and protoplast respectively. The growth of plant cells in vitro is an asexual process involving only mitotic division of cells. Thus, culturing of cells is the method to clone a particular genotype. It is therefore methods it is therefore expected that the plants arising from a given tissue culture should be the exact copies of the parental plant. The occurrence of phenotypic variations among the regenerated plants has been known for several years. These variations were earlier dismissed as tissue culture artifacts. The term somaclonal variations was first used by the scientists named Larkin and Scowcraft in 1981 for variations arising due to culture of cells, that is variability generated by tissue culture. This term tissue culture is now universally accepted. The explant used in tissue culture may come from any part of the plant organs or cells. These include leaves, roots, protoplast, microspores, and embryos. Somaclonal variations are reported in all types of plant tissue culture. In recent years, the term gametoclonal variation is used for the variations observed in the regenerated plants from gametic cells. That means, example, anther cultures like this. For the plants obtained from protoplast cultures, protoclonal variations is used. Now, let's know about the basics of somaclonal variation. Somaclonal variation occur as a result of genetic heterogeneity, genetic. That means change in chromosome number or in structure in plant tissue cultures. This may be due to expression of chromosomal mosaicism or genetic disorders, spontaneous mutations due to cultured conditions. The genetic changes associated with somaclonal variations include polyploidy, aneuploidy variations, chromosomal breakage, deletion, translocation, and gene amplifications, besides several mutations. In fact, the presence of several chromosomal aberrations, reciprocal translocations, deletions, inversions, chromosomal breakage, multicentric acentric fragments have been found among the clones of barley, garlic, and oat. The occurrence of mutations in cultures is relatively low. Mutations may be due to varied nutrients, culture conditions, and mutagenic effects of metabolic products that accumulate in the medium. Somoclonal variations due to transposable elements, mitotic crossing over, and changes in the cytoplasmic genome have also been reported. Now let's know about the nomenclature of somaclones. The somaclones that are regenerated from tissue cultures directly are regarded as R0 or R plants. The cell-fertilized progeny of R0 plants represent R1 plants, R2 plants, R3, R4 like this. The plants are the subsequent generations. Some workers use other nomenclature like somaclones, SC1 equals to R0, SC2, SC3, SC4, etc. for subsequent generations. Now let's know about the isolation of somaclonal variants. There are two procedures commonly used for obtaining the crop plants with somaclonal variations. First is without in vitro selection, second is with in vitro selection. Now first we should know about without in vitro selection. An explant, that means leaf, stem, root like this etc. is cultured on a suitable medium supplemented with growth regulators. The unorganized callus and cells do not contain any selective agent, that is toxic or inhibitory substances. These cultures are normally subcultured and transferred to soot induction medium for regeneration of plants. The so produced plants are grown in pots, transferred to field and analyzed for somaclonal variations. Somaclonal variations of several crops have been successfully obtained by this approach. Let's know about some plants that are nowadays used. Example: sugarcane, potato, tomato, cereals like this. A selected list of disease-resistant crop plants obtained from somaclonal variation without in vitro selection along with pathogenic organisms we will now discuss. Now let's know about the limitation of without in vitro selection approach. There is no directed or specific approach for the isolation of somaclones without in vitro selection. Consequently, the appearance of a desired trait is purely by chance. Further, this procedure is time consuming and requires screening of many plants. Now let's know about the with in vitro selection. Isolation of somaclones with in vitro selection method basically involves handling of plant cells in cultures, protoplast, callus, etc., microorganisms and selection of biochemical mutants. The cell lines are screened from plant cultures for their ability to survive in the presence of a toxic or inhibitory substance in the medium or under conditions of environmental stress. Let's know about some examples of disease-resistant crop plants obtained from somaclonal variations without in vitro selection along with the pathogenic organisms. First, I'll tell the crop name and then pathogenic organisms. First is rice. Its pathogenic organism is Helminthosporium oryza. Maize, Helminthosporium madis. Barley, it's a Rhynchosporium secalis. Sugarcane, Puccinia melanocephala, Sclerospora sachari. Helminthosporium sakari and Fiji virus. Now come to potato. Potato, it's a Streptomyces scabi, Alternaria solani, Phytophthora infestans, potato virus X and Y. Now come to tomato. Tomato, Pseudomonas solanacearum, serum, Fusarium oxysporum. In tobacco plants, Phytophthora parasitica. In apple, Phytophthora cactorum. In banana, Fusarium oxysporum. Lettuce mosaic virus. And alfalfa, that is in the disease is called Fusarium solani. Okay, now let's know in details. The differentiated callus obtained from an explant is exposed in the medium to inhibitors like toxins, antibiotics, amino acids, analogs. Selection cycles is carried out to isolate the tolerant callus cultures and these calli are regenerated into plants. The plants obtained are in vitro screened against the toxin. The plants resistant to the toxin are selected and grown further by vegetative propagation or self-pollination. The subsequent generations are analyzed for disease-resistant plants against the specific pathogenic organism. Besides the disease, Besides the disease-resistant plants, plants with herbicide-resistant and antibiotic-resistant have also been developed with in vitro selection approach. Now let's know about the environmental stress tolerance. High salt concentration in the soil is a major constant limiting the crop development and yield. Many plants with salt tolerance have been developed, example rice, tobacco. Several attempts are being made by plant biotechnologists to develop cold tolerant crops, although the success rate has been very limited. Now let's know about the advantages of in vitro selection approach. The major advantage of with in vitro selection method is the specific selection of desired trait rather than a general variation found at the plant level. This procedure is less time consuming when compared with without in vitro selection approach. Now let's know about the factors affecting production of somaclonal variations. Some of the important factors that influence development of somaclonal variations by without in vitro selection and with in vitro selection. Now we'll discuss. First is genotype and explant source. The nature of genotype of the plants influences the frequency of regeneration and frequency of production of somaclones. Explants can be taken from any part of the plant leaves, roots, internodes, ovaries, etc. The source of explant is very critical for somaclonal variations. For instance, potato plants regenerated from callus of rachis and petioles are much higher compared to those regenerated from callus to leaves. Now let's know about the duration of cell culture. In general, for many plant cultures, somaclonal variations are higher with increased duration of cultures. For example, it is reported that genetic variability increased in tobacco protoplast from 1.5 to 6% by doubling the duration of cultures. Now, let's know about the growth hormone effects. The plant growth regulators in the medium will influence the karyotypic alterations in cultured cells and therefore development of somaclones. Growth hormones such as 2,4-dichlorophenoxyacetic acid 2,4-D in short 2,4-D and naphthalenic acetic acid that is in short NAA are frequently used to achieve chromosomal variability. Besides the factors now we discussed selection of somaclones with in vitro selection are dependent on some parameters. Let's know about the parameters. First point is selection propagule, that means cells, protoplast, cali or callus. Selection agent, that is toxin, herbicide, amino acid analog. Technique used for selection: stability of resistant substance, in vivo testing procedure, ability for regeneration of plants. Now let's know about the applications of somaclonal variations. Somaclonal variations are highly useful in plant breeding programs. The genetic variations with desirable besides the existing preferable characters can be introduced into the plants. In general, the methodology adapted for induction of somaclonal variation are simpler and easier compared to recombinant DNA technology. Hence, they are preferred by some workers. Now, let's know about the production of agronomically useful plants. Production of agronomically useful plants. As a result of somaclonal variation, several novel variants of existing crops have been developed. Example: pure thornless blackberries. Somaclonal variations in a selected list of crops with useful and improved morphological characters now are given. The crops include rice, wheat, maize, sugarcane, potato, carrot, etc. Now let's know about the resistance to disease. So monoclonal variations have largely contributed towards the development of disease resistance in many crops. Example: rice, wheat, maize, sugarcane, tobacco, apple, tomato. Selected crops, monoclonal variants with increased disease resistance developed without the variation selection and with in vitro selection are now we'll discuss. Let's know about resistance to antibiotic stresses. It has been possible to develop biochemical mutants with abiotic stress resistant. Freezing tolerance, example wheat. Salt tolerance, example rice, maize, tobacco. Aluminum tolerance, that is carrot, sorghum, tomato. Resistant to herbicides. Certain somaclonal variants with herbicide resistance have been developed. Tobacco resistant to glyphosate, sulfonylurea and picloram. Carrot resistant to glyphosate, lotus resistant to uh, 2,4-dichlorophenolic acetic acid 2,2-D. Now let's know about the improved seed quality. A new variety of Lathera sativa seeds with a low content of neurotoxin has been developed through somaclonal variations. Now let's know about the limitations of somaclonal variation. Despite several applications of the somaclonal variation, there are certain limitations or disadvantages. Let's know about the disadvantages. First point, most of the somaclonal variations may not be useful. The variations occur in an unpredictable and uncontrolled manner. Next point is many times the genetic traits obtained by somaclonal variations are not suitable and heritable. Next point is somaclonal variations are cultivar dependent, which is frequently a time consuming process. Somaclones can be produced in only those species which regenerate to complete plants. Many cell lines, Cali, may not exhibit regeneration potential. Now let's know about the gametoclonal variations. The variations obtained while culturing the gamete cells are regarded as gametoclonal variations. This is in contrast to the somaclonal variations detected in the cultures of somatic tissues. The term gametoclones is used for the products of gametoclonal variations. As the somatic cells divide by mitosis, the genetic material is equally distributed to the daughter cells. In contrast, the gametes, being the products of meiosis, possess only half of the parental cell genetic material. The gametoclonal variations differ from somaclonal variations by three distinct features. First is mutants obtained from gametoclonal variations give rise to haploid plants since a single set chromosomes are present. Next point is meiotic crossing over is the recombination process observed in gametoclonal variations. Next point is the gametoclones can be stabilized by doubling the chromosome number. Now let's know about the production of gametoclones. Gametoclones can be developed by culturing male or female gametic cells. The cultures of anthers or isolated microspores are widely used. Now let's know about the sources of gametoclonal variations. There are three sources of genetic variations in gametoclones. First is cell culture technique may induce genetic variations. Variations may be induced while doubling the haploid chromosomes. Third point is genetic variation may occur due to heterozygosity of diploids. Finally finished somaclonal variation. Any questions regarding somaclonal variation you can ask me in the comment box. I'll clear all the doubts and subscribe my channel to know more topics about biotechnology and different topics. Thank you.